Hello everyone, and welcome to today's live broadcast and our first keynote presentation, Variant Interpretation to Molecular Diagnostics, Challenges and Knowledge Gaps, presented by Dr. Robert Nussbaum. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're excited to bring you this educational webinar. It's presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. Here's how this presentation will work today. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we'll make sure to follow up with you by email. You can also enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you cannot hear or see this presentation clearly, let us know by clicking the support button at the top right or the Q&A button at the lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your web page and follow the process for obtaining your credits. I'd now like to introduce our keynote speaker, Dr. Robert Nussbaum. Dr. Nussbaum is currently the Chief Medical Officer of Invitae, a genetic information and diagnostic company. From 2006 to 2015, he was the Holy Smith Professor of Medicine at UC San Francisco, Chief of the Division of Genomic Medicine, and Director of the Genomic Medicine Initiative at University of California, San Francisco. He came to UCSF in 2006 from the Division of Intramural Research of the National Human Genome Research Institute. He received his training in medicine in the Harvard-MIT Joint Program in Health Technology, his internal medicine training at Barnes Hospital, Washington University, and his genetic training at Baylor College of Medicine. He is board certified in internal medicine, clinical genetics, and clinical molecular genetics. He is the co-author of over 200 peer-reviewed publications on a broad range of topics in basic applied human genetics, and is the co-author with Dr. Roderick McKenzie and Huntington Willard of the popular textbook of human genetics, Thompson and Thompson, Genetics in Medicine. Dr. Nussbaum was one of the board American Society of Human Genetics, where he has also served as president. He was elected to the National Academy of Medicine in 2004 and the American Academy of Arts and Science in 2015. I'd now like to turn the presentation over to you, sir, Dr. Nussbaum.
Valdez, thank you very much. It's a great pleasure to be here this morning. I'm going to be talking to you about integrating genetic testing into mainstream medicine. Whole genome and whole exome sequencing. What is the technology and why is it so revolutionary? Does this technology solve the problem of integrating genetics into mainstream medicine? What I'm going to be showing you today is that this new technology has made an enormous difference in making it possible to integrate genetics more formally into mainstream medicine, but there's still a lot more to go that is not just the technology of sequencing. This is an entire ecosystem that needs to be brought into play in order to make this happen. Before we had next generation sequencing, how did we do sequencing? We essentially did one gene at a time. So for example, you might start with some DNA, and you'd have a gene of interest shown in red here. And you would look at these fragments and then use polymerase chain reaction to amplify the particular sequence of interest, shown here with the red arrows. You would then have essentially a test tube full of these sequences that are amplified, and then you could take this amplified fragment of DNA, sequence it, and compare it to what we call the reference sequence or the reference genome. So what is the reference genome? Whose genome is normal? Well, nobody's genome is normal. There are billions of different genomes on the planet. We therefore need a consensus genome to which we can compare an individual sequence. And the reference is really a consensus of the most common sequences found in volunteers. It is not currently very ethnically balanced. There are certain areas, geographical areas of the world that are underrepresented in the consensus. And we're learning more and more as we go along as to what kind of variation is common and, in essence, normal, does not cause disease in many other parts of the world. But we have a long way to go until we have a reference sequence that is really a consensus of the world's population. So whole genome analysis, which is either whole genome sequencing or whole exome sequencing, whole genome sequencing is what it says. You obtain the sequence of most of the 6 billion base pairs of DNA in any individual, while whole exome sequencing is containing the sequence of almost all of the 2% of the genome that contains the exons of the approximately 20,000 or 24,000 genes, depending on how you want to count them, that encode proteins. And that is what's called the exome. Next generation sequencing is also called massively parallel sequencing. And the reason is because the most widely used next generation sequencing technology provides a few hundred base pairs of sequence from many millions of DNA fragments, enough to cover the whole genome at an average of 40 to 60 times over, at the same time, in parallel, in a single flow cell. So what that allows you to do then is exactly what it says, which is in a parallel manner sequence massively, many fragments at the same time, therefore being able to generate billions of base pairs of DNA, which you can then line up and examine for changes or differences between the sequence of that individual and the sequence of the consensus. So here's ideal whole genome sequencing, which is where I hope we'll be going over the next few years, but it's going to take some technological advances to make this happen. So you start with DNA, and you've got a series of fragments from the DNA that contains some sequences of interest. For example, in orange, what I show you are the exons of gene 2, and in brown, the exons of gene 1. So you take these fragments and you carry out next generation sequencing so that you get a bunch of partially overlapping sequences, which you then line up by matching the bases so that they form a contiguous or contig sequence. And then you assemble that de novo into some sort of contiguous sequence that combines all of the sequence in a linear fashion. This would be ideal. Unfortunately, we can't really do this very well. And the reason is because the sequencing runs are relatively short. They're only about 200 to 300 base pairs currently for the most widely used next-generation sequencing platform. There are others that will generate much longer reads, and there are other technologies coming along that will allow you to do much longer reads. And that, I think, will get us closer and closer to ideal whole genome sequencing. But currently, for most whole genome sequencing, because the reads are relatively short, you run into a problem. And the problem are repetitive DNAs. For example, ALU, which makes up about 11% of the human genome here, and LINE, which makes up about 20% of the human genome, they are inserted throughout the genome. So that when you take a DNA and generate fragments and do next generation sequencing, you may line up the nearly or actual identical sequence from repeats that are located in many different parts of the genome. And therefore, this is not a true alignment. You're mixing and matching. So what's on blue, which are the unique sequences that are next to the repeated sequence, these are actually not contiguous in the genome. They could be from completely different chromosomes. And so it's very difficult to assemble when you've got repetitive DNA of this kind. So instead, to do actual whole genome sequence, once again, one needs to rely on a consensus as a scaffold upon which one applies the sequence you generate. So you carry out next generation sequencing, and the brown is the from gene 1, let's say exon 2 from gene 1, and orange is exon 3 from gene 2. You generate the fragments the way we talked about, and then line them up against the reference sequence and so that you can form them into a contig. And the repetitive DNA is simply not looked at in great detail if it is longer than you can bridge in a sequencing run, sequencing fragment. Whole exome sequencing is similar, except that there's an additional biochemical step. There are a number of different ways to do this biochemical step. I'm just going to show you one example. So once again, you've got DNA, you've got fragments that contain, let's say, fragments from exon 1, exon 2, and exon 3 of a gene, shown in brown, pink, and orange. What you then do is you have a mixture of short synthetic DNAs, oligonucleotides, that are complementary, will base pair to the exons of interest, shown up here, and you attach these short fragments of DNA to magnetic beads. You then mix these together, and you allow the short complementary oligonucleotides to anneal to base pair with the sequences of interest, and then you use a magnet to pull the magnetic beads out containing the fragments of interest. And then you can elute the fragments off from the beads right here, and now sequence them, and what you generate then are sequences of the various different exons. Shown here is the exon 3, and then align it once again to the reference to form to the consensus, so that you know that you're looking at a particular region of the genome by using the consensus as a scaffold. So that's the basic technology that's currently in use, but as I want to just repeat that there are many ways that people are attempting to improve the next generation sequencing. There are different methodologies for sequencing. There are different ways of trying to generate much longer reads, in particular reads that will go across and will bridge across some of the repetitive DNA, so that you can actually do a de novo alignment, or I should say a build, because you will actually have unique sequence anchoring the fragments that you are sequencing at the time. In any case, we'll go on now to talk about what are the 4 major steps that any sequencing technology really needs to meet, or standards they need to meet if the sequencing technology is going to be used in clinical medicine. And there's something called the ACE framework, ACCE, which was created by the Centers for Disease Control Office of Public Genomics. This is from Dr. Muin Khoury and all of his colleagues there. They've done a very nice job of in a simple way, thinking through what are the basic requirements for sequencing technology to be successful in the clinical sphere. The first, and this is true, of course, of any test, not just sequencing, but I'm going to be applying it specifically to sequencing here. The first is what we call analytic validity. Can you get the right result from the right patient? And this can be anything from something as simple as making sure that you haven't swapped samples, that you don't have a mislabeled sample, something very basic, to something much more technical, which is if there is a complicated variant, a complicated change in the DNA sequence, is the technology capable of detecting it? The second is clinical validity, and that is, given that you can find what is the variant in the genome, what does it mean? Does it have any association with, or does it increase the risk for any disease? Does it have any health implications at all? And that is in some ways what we talk about as the penetrance in genetics, which is, given that you have a particular genotype, what is the likelihood that it's going to affect your health? What we call the phenotype, the outward manifestation, phenotype, outward manifestation of an underlying genetic alteration, which is what we call the genotype. Assuming we can interpret these variants properly, the next step is, okay, well, how useful is it to know that a person carries these variants? And do they make a difference? And if so, make a difference to whom? And we'll talk about that later. And then finally, is there value to society beyond just the individual in having test results? And why is that important? Well, it's important for ethical reasons, certainly, to be applying a technology which could have important ethical ramifications in society, but there are also economic and other social implications, depending on what country you're living in, some of these tests, maybe most of these tests, may actually be paid for by tax dollars or by government funds, in which case the government really has a role to play in deciding whether they wish to pay for having these tests done. So are there economic and then legal and social reasons? Would these tests be used, for example, in deciding whether people can get health insurance or life insurance? Or would they be used in the workplace to make decisions about whether someone can or cannot be employed? These are all important implications that have to be considered when thinking of applying sequencing to human beings. So let's talk about analytic validity. First, what we're really talking about here are true and false positives. Did you do the right test on the right patient, and did you find an abnormality when it's there and not when it's not? Another way to think about that is the term sensitivity and specificity. In people at risk or with a disease, how often is the test or the sequence positive? And specificity, in people at risk or without the disease, who are not at risk or without the disease, how often is the test negative? Sorry, there's a misprint in this slide. It should be in people not at risk or without the disease, how often is the test negative? And that's the specificity. Now, in whole genome sequencing or whole exome sequencing or panel sequencing, where you're looking at a number of genes at the same time using next-generation sequencing, part of the issue about sensitivity is a random effect of coverage. By coverage, what I mean is, if you remember back to when I showed you how to do this, how the sequencing is done, you line up sequence reads and then apply them to the reference genome so that at any particular base, you can count how many reads, how many unique reads did you actually generate from that person's sample. And that's called the coverage per base. How many independent reads contain that base? And so here's a curve of what might be the distribution of coverage per base, where the maximum is around 350 or 400 fold around a particular base. And if you accept that somewhere around 30x coverage is the threshold below which you might not be comfortable that you can actually call a particular base accurately, that you need to see at least 30, if not more, reads containing that base in order to be confident that you can read that base properly. You can see that if you have an average of 350 or 400 coverage, that most of the time, the vast majority of the time, there is no problem with having more than 30x coverage. You can see even down here in the left-hand tail, you're way above 30x coverage. However, if your coverage is only averaging less than 100x, or maybe down around 60x or 80x, then you have a problem, because part of this tail is actually below 30x, perhaps somewhere between 2 and 3%. And these numbers vary. These are not absolute numbers. It depends a lot on a number of technical factors. But the basic concept is that there are random effects of coverage on your sensitivity, on your ability to read a base at a particular location. There are also non-random effects on exon coverage. So here's an example. Here's a picture that I obtained courtesy of Dr. Sarah Garcia, where you have shown in green and shown in blue the coverage per exon in a gene. So each of these bars is an exon. So here's exon 1. Right here is exon 1. And here's exon 2. And here's exon 3. Okay? And what you can see is that in blue is the average depth of coverage per exon. So you can see that exon 1, there are close to 75 or even 80 coverage of that exon. Whereas for exon 2, it's a little lower. It's more like about 40. And exon 3, the vast majority of that exon, is there are less than 10 to 15 reads per base. And so that under this circumstance, exon 3 might be very hard to read. And why are there such differences between different exons? It's because the coverage, number of reads per base, is not just a random effect. There are also non-random effects that are based on the base composition of the exon, GC coverage, GC content, for example. Also, the type of sequence there, if there are runs of the same base, it might be difficult to capture. Because remember, when you're doing an exome, you're doing a biochemical step of pulling down the approximately 2% of the sequence that is contained in exons. So there may be non-random effects as well in terms of how well you capture particular exons, etc. You can go in and deliberately improve the coverage for bases, for exons that are poorly covered. So what's shown here, for example, is you modify the chemistry or you modify the baits that you're using. So exon 3 now has been boosted up to almost 30x by deliberately going in and making a point of trying to pull down more of exon 3 than before. And you can see that across the gene there are marked improvements, and yet still, even with the use of the extra baits, there are still exons that are not being well covered, shown there and there. So in addition to random and non-random effects for analytic validity, for being able to detect what's there, it also depends on what kind of variants. Single nucleotide variants, or SNVs, which is a single change from one base to the other, are probably the easiest to see if you've got decent coverage. But there are some biases, some difficulties in being able to detect variants other than SNVs. For example, copy number variants, where you've got large pieces of DNA, perhaps a few hundred to a few thousand base pairs, that will simply be missing from the sequence. And because the sequence reads are so short, it's very difficult to know that they're missing. But there are approaches to being able to do this, which I'll talk about in a minute, but that still represents a challenge. So the kind of variant you're trying to find also has an effect on analytic validity. So, for example, this is data from my colleague, colleague Stephen Lincoln. There are many pathogenic variants that are quite technically challenging for next-generation sequencing. Here's an example: hereditary breast and ovarian cancer cases, over 1,000 such patients. And pathogenic and likely pathogenic variants were found in 260 out of the over 1,000. And here's the type of variants. So only about a third were single nucleotide variants, whereas the small indels, which are short repeats, were a little over, short insertions or deletions of sequence were more than half. And what you've got is an even greater array of complicated variants here. 4.6% were multi-exon deletion duplications, 3.8% were deletions and duplications of a single exon within these genes, large insertions and deletions, 3.5%, and then complex rearrangements, including inversions, 1.5%. So SNVs, which are the simple variants to find, are only about a third, and pathogenic variants are particularly enriched for such technically challenging such variants. I mean, if you look across a genome and just ask how many differences are there in a genome between any individual and the consensus, you'll find between 3 and 4 million, and they are by far the most common difference. However, many of them are common in the populations, many of them are benign, whereas if one's actually trying to find the variants that are causing disease, they have a higher proportion of technically challenging variants to find than just the total variation between any individual and the consensus. So 13.4% are technically challenging. And here's some examples of pathogenic disease-causing variants in patients that one would call hard to call by exon sequencing. So, for example, there's a novel ALU insertion, a very large indel. This is involving well over 126 bases that have been deleted. A splice site But unfortunately, this splice change is right next to a run of 25 base pairs of homopolymer, just A's, 25 A's in a row. And next generation sequencing has difficulty detecting that A to T change when it's sitting right next to runs of a bunch of A's. An inversion involving exons 1 through 7. A tandem duplication in a 5' CPG island, which is very CG rich, as where its name comes from. Or you can have pseudogenes, such as in the gene PMS2, which is involved in hereditary colon cancer, Lynch syndrome, where exons 11 through 15 are almost perfectly duplicated within a pseudogene that's located elsewhere from the real gene. It's not the real gene itself, it's a pseudogene. And so that you may get reads from the pseudogene section that will fool the software that's trying to do the alignment, and you'll end up overcalling mutations that are actually not in the real gene, they're in the pseudogene. So the prevalence out in other areas of genetic disease, not just in hereditary cancer. So it's about 8.1%, these pathogenic deletion duplication events. They were seen in a little over 1% of all the patients tested in the lab where I work, but 7.8% of all patients with any pathogenic variant. And it varies from a clinical area. So 8.1% in hereditary cancer, less in cardiology, much more in neurology because of major alterations that occur in the Charcot-Marie Tooth, PNP22 gene, and in pediatric genetics in general, almost 7%. So these difficult to see variants are a challenge to analytic validity, and yet they're absolutely essential that one be able to see them if you're going to have a good sensitivity for detecting pathogenic variants in patients who need a diagnosis. Now, for copy number variants, although they may be hard to see, if you have sufficient coverage and you control the lab processes very carefully, it is possible to get a very good idea of whether there's copy number variation between the individual you're testing and the consensus. So here's an example of 3 patients where what we've done is we've mapped out the read depth, the number of sequences that are seen over exon 1, over exon 2, and exon 3. And you can see that it's really, they're very tight, they're very close to each other. The total number of reads varies from somewhere around 500, give or take 50, to a little over 200, give or take 20 or 50. And here you can see that you're up around an average of about 325. So these are reasonably tight, and that is in contrast to this situation, where you have a patient who has a deletion in exons 1 and 2, and the actual number of reads seen is reduced significantly, in half. You can see that here, whereas exon 3 is intact in this individual, and so they have the same number of reads as what's seen in the controls. You can also do what we call split read analysis, where if the deleted region happens to include 2 exons that are being looked at for exon sequencing, you may generate a fragment here, right here, that contains the junction between this exon and this exon with the deleted region missing between them. And this would be what we call a split read, that you have a single read that jumps from this from the exon here to the exon here within a single read, and allows you to infer that there's been a deleted area between them. That's assuming, of course, that this is all unique sequence and you're not being confused by any repetitive DNA. So there are del-dup methods that are powerful when used in combination for being able to detect larger deletion duplication events. So there are limitations of the current next generation sequencing. The sequencing reads are reasonably short, 200, 250, maybe 300. The technology is changing very rapidly. Don't hold me to these numbers, but these are the general ballpark figure. NGS does not allow de novo construction of individual sequence because of repetitive DNA. So you really have to compare the unique part of each individual sequence to a standard, quote, normal, which is actually a reference or consensus sequence. De novo assembly of contiguous sequence across repetitive DNA sequences and segments is difficult. Repetitive DNA is a real challenge, including some very medically important regions, such as triplet repeats in the Huntington disease, and direct detecting deletions or duplications in this range of 100 to 500 base pairs still represents a challenge because of issues with sampling and coverage. So we'll go to the next step now, which is clinical validity. How do you know that what you find means anything? So clinical validity is in a people who are positive for the test, is the disease present or is there increased risk? This is the opposite of sensitivity, which is in people who have the disease or have an increased risk, how often is the test positive? Positive value is the opposite. In people who are positive for the test, how often is the disease present or increased risk? In other words, it's prediction. And negative predictive value is in what in medicine we call the rule out. And that is in people who are negative for the test, how often is the disease absent or is the risk not any greater than that of the general population? Now, when you compare an individual's whole genome sequence to a reference, what do you find? These are ballpark numbers, once again. They're not meant to be actually true for any one individual. But in general, you see approximately 3.5 million single nucleotide variants and about 500,000 insertions and deletions. And if you go which ones are within genes, which ones are within the exons of genes, we're down to 47,000 for single nucleotide variants and almost 6,000 insertion deletions in exons and on down. So you can see that there's a lot of variation. Encoding exons alone, if you just leave out the 5' and 3' untranslated regions, you still have 20,000 single nucleotide variants and close to 500 insertions or deletions. But remember that insertions and deletions are overrepresented as pathogenic. And so these are very, very important to be able to detect. But you still see a lot. And so the question is, when you find a variant, how do you know that that variant is actually involved in causing disease? Because there's this background of a large number of variants that you're going to find anytime you sequence anybody. So if you're going to do variant interpretation, there are two steps. One is, if you find a variant in a gene, is that gene known to be involved in a particular disease? In other words, the gene-disease relationship. And if that gene is known to be involved, is there evidence that the variant, the particular change you found in that gene, is related to causing disease? So for gene-disease relationship, this is an approach that's taken by the ClinGen project, which is a large NIH-funded project in the United States, attempting to establish the lines of evidence that would allow one to infer that there is a relationship between changes in a gene and a particular disorder. So inside, what you got here is definitive evidence or strong evidence. And you need that if you're going to be doing predictive testing or for incidental findings, where the patient has no reason to think they might have a change in that gene. They actually don't have a disorder. They're actually trying to find out if they're at risk for it, or if you stumble on a change in the gene while doing a test for some other indication. Whereas if you're doing a diagnostic panel, less stringent evidence is needed because the patient is already demonstrating the phenotype. And then finally, if you're doing exome or genome, where you're actually trying not only to find variants in genes, you're actually trying to discover new genes that were not known before to be associated with a particular disease. There, more limited evidence or disputed evidence can be brought to bear in trying to infer that a particular change is in a gene, that gene in which the change occurs is associated with the disease. So the point I'm trying to make here is that the evidence you need to include a gene in a clinical report depends on the context, depends on what the patient is presenting with, what sort of testing is being done, what the purpose of the test is. Now, in addition to gene-disease relationships, Once you find that a gene is, that changes in a gene can be associated with disease, how do you know whether any particular variant you find in that gene is actually associated? And the American College of Medical Genetics, with the Association of Molecular Pathology in the United States, have put together a framework for 5 categories of variant interpretation. At the very top is that one is quite certain that that variant is responsible for disease. We call that a pathogenic variant. And at the opposite extreme is variants that we are quite certain have no impact on health, do not cause disease. We call those benign. In between are various levels. So we have the term likely pathogenic, and I'll tell you about that in a minute, what that means, and likely benign, meaning we're not 100% certain or 99% plus certain that it is benign or it's pathogenic, but we're leaning very heavily that way. And then finally what's left is this category in the middle, which are called variants of uncertain significance, or VUSs, where it's a change and we actually don't know what it means. It can be, it could end up being benign, and in fact, our current experience is most of the time they do end up being benign. Somewhere around 80 to 90% of the time, VUSs, when they get reclassified, are reclassified as benign. But it's not always the case. What is the meaning of likely? Likely is somewhere around 90% plus, whereas being pathogenic or being benign, for sure, is more like 99%. So this is the framework that was put together. It's a complicated slide. I don't want to go through it all the way, but what I just want to point out to you is the following. Across the top are levels of evidence that one would consider to be either strong or supporting for benign, or levels of evidence that one is considered to be either supporting, moderate, strong, or very strong for being pathogenic. Okay? And so, for example, let's look at population data. Over here, if the minor allele frequency, or MAF, is too high for the disorder, or it's observed in controls, and that's inconsistent with the penetrance disease. In other words, there are too many people who have this variant who don't have the disease. And exactly how you measure too many is something that we would have to discuss in more detail. But that's the basic idea. It's just too common to be responsible for disease, since it's found so often in people who don't have disease. At the opposite end, if it's found in affected individuals statistically significantly more frequently over controls, then that would be considered to be strong evidence for being pathogenic. So these are the types of evidence that are used in this process. One has population data. We have computational data, where you look at the change itself and try to infer, for example, using conservation. Is this an amino acid that Mother Nature has conserved over hundreds of millions of years, or is it one that it appears that it can wobble a lot? It can be a number of different amino acids, and it doesn't matter. Functional data, have people actually done experiments to show that this variant affects the function of an enzyme or changes the function of a cell? Does it segregate in families? Does it follow? So does the disease and does the variant, are they co-inherited? Do they, in other words, segregate down through a family? Is it found as a de novo change, as a new mutation in an individual with the disease, not present in either unaffected parent, etc.? So these are the sorts of evidence that we try to put together, and using databases of various kinds. And what do people say about this variant? Has it been seen by a number of different laboratories, and has it been seen by researchers? And how do we collect that information? And I'll be talking about that in a minute. But that's the kind of data, and then you can assess how significant it is by assigning a strength from strong to supporting for either being on the benign side or being on the pathogenic side. And what you do is then add up how many very strong or strong pieces of evidence you have. What does it take to consider a particular variant pathogenic? Well, what you can see here is that what it takes to be pathogenic is you need one very strong and one strong or two or more moderate or supporting piece of data, or two strongs, or one strong and et cetera. So I won't go through all this, but basically, if you cannot place a variant into any of these categories between benign, likely benign, up to pathogenic, then you're left with, if the criteria are met or there is discrepancy, that there's piece of evidence to point you one way, another piece of evidence to point you the other way, then it's called a variant of uncertain significance. We just don't know what it means. So one of the key issues is if you apply this sort of information, and a number of different laboratories apply it, what happens? So this was an interesting experiment, and this is data I got from Dr. Gail Jarvik. I'm very grateful to her for sharing this with me. It's from the Clinical Sequencing Exploratory Research Group, the CSER group, where they did a bake-off, basically. They gave 99 variants to 9 labs, and they asked them to classify them according to their own internal lab methods, but then also adhere as strictly as possible to the ACMG guidelines, which I just showed you. And then what they looked at was both intra-laboratory, within a laboratory, and inter-laboratory differences in variant classifications. And the results were really quite interesting. Along the diagonal here was perfect match between what people said using their own lab information, which is across the top, lab class, and then what they would have called it if they adhered to the ACMG guidelines as strictly as possible. And you can see that there was a pretty good alignment, 347, but there were still a number of variants where there were discrepancies off the diagonal. So, for example, 2 labs classified this as pathogenic when they used their own classification, but when they applied the ACMG classification, those 2 variants actually ended up being called VUSs. So it turned out that there was 79% identity, but that means 21% of the time there was a mismatch, and some of them quite significant. So how does one go about figuring out when variant interpretations are discordant? Well, one of the tools that's been developed is essentially a very large crowdsourcing effort called ClinVar, which is an NIH-funded grant that has put together a database where laboratories, researchers, other expert groups can put in variants and their interpretation and the evidence for that interpretation. And ClinVar has been somewhat controversial. I'll call it a hot topic. There's good, there's bad, and there's ugly sorts of information floating around about the value of ClinVar. And a group here at Invitae, led by Steve Lincoln and Shan Yang and Scott Topper and a number of others, have tried to look at this question of when is data concordant and when is it discordant. So for BRCA1 and 2, they did a download of ClinVar in May 2016, and what they looked at was data that met the following criteria, and they tried to make rather objective criteria. For example, it had to be submitted by established clinical labs. It couldn't just be a random researcher. It had to be labs that had put in at least 200 classifications in ClinVar, and they had to be entries that were less than 5 years old, because older, we know, and I'll show you in a minute, older entries have a tendency to be less consistent, and it's because we knew a lot less, and for example, we were lacking one of the most important tools that has become available for variant interpretation in the last few years, and that is a large database of exome variation called the EXAC from Dan MacArthur at the Broad Institute, which has been an extremely valuable database that people use to try to get some idea of what the normal allele frequency is for any particular variant. And they looked at comparisons that would make a difference. In other words, pathogenic and likely pathogenic, which are treated clinically similarly, were put in one bucket, and then benign, likely benign, and VUSs were put in another bucket. So this is what we'd call clinically actionable, and the VUS, likely benign and benign, is what you would call not clinically actionable. So we dichotomized it into those 2 categories. And looked at a number of different laboratories in the United States who had sufficient data to be able to do this sort of analysis. And in general, what you can see here across the top is that there was tremendous consensus or concordance between different submitters, there's really not a lot of very significant discordance, but there was some. I mean, for example, we're talking about discordances down around 97% or 98%. So that means that between 2 and 3% of these BRCA1 or 2 variant interpretations were not totally concordant across all of the laboratories shown here. So what's responsible for the discordance? So we took a very deep dive into ClinVar. We looked at all unique variants from all genes that Invitae offers with at least 2 classifications submitted by established clinical laboratories, including data from the Myriad Genetics, which was submitted via something called the Sharing Clinical Reports Project, where those variants were obtained from clinicians who had ordered the test. It ended up being 38,000 total classifications, 14,800 unique variants, and an average of about 2.5 classifications per variant from about 520 genes. And what you can see here is that, once again, if you dichotomize between pathogenic, likely pathogenic versus non-clinically actionable, VUS, likely benign, benign, and look at different clinical areas, shown in red are the number of classifications that were found, and the consensus shown in red and blue, and the consensus is in the brown diamonds was a more than 2/3 of the interpretations were concordant. So there could still have been discordancies, but 2/3 or more of the labs that submitted them had the same interpretation of clinically actionable versus not. And shown in the blue diamonds here, blue diamonds, was 100% concordance. There was no difference. It was a complete agreement. So you can see that in cardiology, the concordance rate was dropped down to close to 92% for all to agree. So there was more discordance in cardiology than hereditary cancer. In the metabolic area, the total number of variants interpreted was actually quite small, so these numbers are less dependable. This is a very small number. But in cancer and cardiology, there were a lot. And if you looked at pathogenic, likely pathogenic versus VUS versus likely benign, benign. So now instead of dichotomizing, you were to trichotomize. Then the discordances, as you might imagine, grew even more. So in cardiology, for example, to have complete consensus that something is P or LP versus VUS versus LB or B, we're down around 75 or 77%. What's the source of these discordances? Why are these discordances there in ClinVar? What we find is it really has to do with who submitted the variant interpretation. If you separate the variant interpretation submitters into clinical testing laboratories, shown here in green, literature only, or some sort of curation, and for example, the online Mendelian inheritance in man, which has alleles listed as being disease-causing, although OMIM does not interpret those variants. They don't do their own independent variant interpretation. They simply report what's in the literature, and there are databases that do literature reports, versus laboratories that are doing research that submit. You can see that there's a significant difference in variant interpretation by source, with the concordance rates being 99.5% among clinical testing labs, and it drops if you use literature. I think what this is a reflection of is that older literature in particular, the requirements for being concordant was actually quite a bit less. The requirements were not as stringent as they are now. And so older data that was published in the literature that states that a certain variant is or is not pathogenic is not as reliable as current data. So let's just, in the last few minutes, talk about what is one of the most difficult areas, which is clinical utility. Are test results useful to patients? Well, what do we mean by clinical utility? Depending on who you are, like the blind men feeling an elephant, they all think it's something different. Well, different people think of clinical utility differently. The patient says, give me information I can use to make life decisions that I will find personally helpful. A provider will want information that helps with diagnosis, prognosis, or treatment, what we call clinical utility. On the other hand, someone who is paying for this, a payer, will say, I only want to pay for something that the provider is actually going to use to change management. And then finally, there may be a utilization manager, somebody who says, I don't believe in any of this stuff. I only want to pay for something that improves an outcome, preferably proven through a randomized controlled trial, for which there is almost no such data available for the vast majority of genetic testing. If for no other reason, most genetic disease is rare, and putting a randomized controlled trial for whether testing is beneficial or not is simply not very practical. So, what do we mean by clinical utility? The test provides information the patient and the clinician would like to know. A smaller subset is test result that changes management, and even smaller subset is a test that provides information that improves outcome or saves lives. One very nice example, I think, of what has been demonstrated as clinical utility is making a diagnosis and ending the diagnostic odyssey. This is a lovely paper from Journal of American Medical Association from the group at Baylor College of Medicine. We looked at 2,000 consecutive patients referred for whole exome sequencing because nobody knew what was going on with them, undiagnosed disease. About 25% had a diagnosis made by whole exome sequencing. And this shows you, depending on what their presentation was, developmental delay, which is here, or neurological plus other things, or just a neurological disorder, or non-neurological. Red is the number diagnosed, blue is the total, and you can see that there's really a substantial diagnostic yield. 25% of the time they could make a diagnosis, which was not able to be made by any other method. Now, depending on the diagnosis, that then might lead to management changes or might not, but at least from a diagnostic point of view, there's obviously exomes have clinical utility. And what's interesting is 5% of these patients actually had blended phenotype, that they actually had two single gene disorders, which together created a disorder that no one could diagnose because it didn't fit into either one perfectly. So whether something is actionable or not is really in the eye of the beholder. There's a metric that one can put together as to actionability based on things like the severity, what's the nature of the threat to health, how likely is it that this variant is actually going to result in this disorder, how effective is the intervention that you're proposing, in other words, to change the management, the nature of that intervention, how risky, how burdensome, how intensive is it, and what is the level of evidence that supports this actionability. So this is a metric that was put together by the ClinGen group, and I think it's a useful way to think about trying to quantify, and you can see there are scores here, 0 up to 3, as well as scores for the quality of the data for figuring out whether something, how actionable something is. So is this actionable? BRCA1 or BRCA2, this is the making this diagnosis, and this is the risk of developing breast or ovarian cancer with the arrows showing you what the lifetime risk is in the general population. And so knowing that you're at risk for breast cancer, that's upward, your risk is upwards around 60% or more if you carry a BRCA1 lifelong, is likely to be an actionable result, and there are management guidelines that will change the way you deal with patients who have such mutations, offering them prophylactic mastectomies and salpingo-oophorectomies. Another way to think about clinical actionability in this disorder, we have multi-gene panel testing for hereditary breast and ovarian cancer. This was a paper published in JAMA back in 2015, showing that patients who don't have BRCA1 mutations, that there was an increased yield of such patients, so that if you only tested BRCA1 and 2, you missed 3.8% of patients who had an actionable mutation, and of those, about half would get additional disease-specific screening or prevention measures. So one can interpret that as a form of actionability. Although I have to say that most of these management changes are based on expert consensus and are not based, for the most part, on any kind of prospective trial. And so one could argue, is this really actionable if it's a bunch of experts who simply say you ought to add MRI scans, for example, to the breast surveillance? And can you actually demonstrate that that has a benefit? So I'm going to finish up. These are, I'm running out of time here, and I don't want to cut off the discussion. So I think that I'm going to end on this, which is, what are the myths about using genomics to personalize medicine? So I hope what I've showed you is that it is not true that the technology is going to solve all the problems. There are issues of clinical validity and clinical utility that have to be dealt with, no matter how good the technology is. I find an attitude that genomics is really so cool, you don't need to gather the effectiveness data needed for the translation pathway. I think this is not true. People in the field love genomics, but there are people outside the field who need to be convinced, in particular the people who hold the purse strings, that the patient and providers of genomic testing do not get to decide what's useful. This is a myth, and the third-party payers and the governments who support healthcare are delighted to pay for it. This is not the case. We have to make the case, show the evidence, in order to convince the payers to pay for it. So I'm going to end on this quote from Bill Gates. We always overestimate the change that will occur in the next 2 years, and we underestimate the change that will occur in the next 10. Don't let yourself be lulled into inaction. I think we have made tremendous progress in integrating genetics through the use of genomics and next generation sequencing into medicine. We've got more to do. We cannot just stop here. There's a lot more evidence we need to gather and a lot more convincing we need to do. With that, I'm going to thank you very much. Okay?
Dr. Nussbaum, thank you so much for that informative presentation. We want to begin our Q&A segment. So here's a reminder as to how audience members can communicate with us. Questions can be submitted via the Q&A button at the lower left. If we are unable to get to your questions due to time constraints, Dr. Nussbaum will be answering submitted questions via email. We also would like to ask you to take our 2-question poll. Very simple. It will be posted up on the site as we're talking, and we'd appreciate you taking that. So let's begin our Q&A. We have several questions today. One of our audience members would like to know, how does the clinical utility of genetic testing work? Who does it?
Has a number of different, clinical utility really has a number of different origins. I would say the lowest level is when a group of experts get together and say that laboratory result or that sequence variant, knowing that in this patient, is useful because we think it should lead to some change in the way this patient is managed. The next level above that would be that someone actually does some sort of a retrospective look at a group of patients, some of whom had the testing, some of whom didn't, and you could show that if one hypothetically had introduced some difference in surveillance, that one could have had a better outcome for those patients who had been tested versus ones who didn't. And ultimately, some sort of prospective follow-up of patients. It doesn't necessarily have to be a randomized controlled trial, well, that's obviously the most powerful, but some sort of prospective showing if some patients get a change in their management and others do not, based on a genetic test result, that one group of patients does better than others. And that sort of level of information has been shown. Your control can actually be historical. In other words, a group of patients, none of whom got genetic testing, and had a certain outcome. And now you're going to look at a bunch of patients who did get genetic testing, and you're going to change their management based on what you see. So this is evidence development that has to be done by the medical and scientific community. It's not just going to happen spontaneously.
We only have a couple minutes left. We have one audience member that would like to know, WGS is more functional towards diagnosis and more expensive than WES. How do we use effective molecular diagnosis in continually expensive healthcare management?
I'm very much of the opinion that that we need to match the particular technology and the cost of that technology versus the yield. So, for example, I can easily see situations where we know that there are, let's say, 250 particular genes that are responsible for a particular condition, let's say childhood epilepsy. To do a panel of those 250 genes, a full clinical report, costs a fraction of what it would cost to do a whole genome sequence and do a clinical report on the sequence. I should mention, don't be fooled that the only cost of a test is what it takes to run a sequence. You also have to analyze that sequence and give a full clinical report, and that adds significantly to the cost. So I think some sort of staged approach where you use a much less expensive test first and see if it answers the question. And if not, then in a fraction of patients in whom you don't get an answer to the question, move on to the more expensive testing. I think in our current situation, that makes sense. I think in the long run, what's going to happen is that whole genome sequencing will become the standard for all genetic testing when the cost of doing it and the cost of analyzing it drops to the point that makes it practical. We're not there yet, but I could imagine that we could be in a few years. So I think we're going to.
Thank you, sir. We have time for one more question, and then we will have to close this presentation. What do you see as the future of variant interpretation? And if you want to add any closing remarks, that would be fabulous.
The future of variant interpretation is going to be multifaceted. First, just as we do more and more sequencing and bring to bear information about the patients who are being sequenced through things like the big project in the United Kingdom or the personalized medicine project in the United States, we are going to be connecting people's health records and their sequence much more closely and in much larger numbers. And this is going to allow us to make better inferences about what different variants mean for health. I mean, at this point, we're really very much at the early stages of this, where we're looking at changes in genes that have major effect, things that cause disorders that are inherited according to the Mendelian manner. We are going to have to expand to the point where we can look at multiple variations in multiple genes and how that then affects health. That's going to take very large number of patients and much, much larger amounts of data. So I think the future of variant interpretation is in large data collection and large data assessment. The other is I think we're going to get better and better at being able to make in silico interpretations. We're really at the infancy of that as well, to be able to look at a sequence change, and because we know a lot about the particular protein or that particular gene, we can make a very accurate, or at least contribute a very accurate piece of information to variant interpretation. And then thirdly, as these databases of variants that are linked to particular disorders increase, things like ClinVar, they are growing at leaps and bounds. We've got hundreds of thousands of variants that are in ClinVar. I would expect it to grow into the millions, and that's going to provide also a very powerful large data set when used properly. So I would say the future of variant interpretation is going to be large data sets, better phenotype information, and better in silico assessment of variation and their impact on proteins.
Thank you again for this fabulous presentation, Dr. Nussbaum. I'd like to also take the time to thank LabRoots for making today's educational webcast possible. Today's webcast will be available for on-demand through July 2017. Keep an eye out for an email from LabRoots alerting you when the webcast will be available for replay. We want to also encourage you to forward the announcement to your colleagues who may have missed today's live event. Thank you for joining us, and we hope to see you next time. Goodbye.